Welcome to Noble Innovation, where we delve into the inspiring stories of remarkable individuals whose impact has changed the world for the better. Noble Innovation is a play on words for Nobel Prize, as we begin with a series with those who have been honored with the Nobel Prize. A major goal of this podcast is to inspire you to add more innovation in your life. We'll focus on the paradigm-shifting aspects of these impressive innovators and try to glean new ways of thinking about life that might help us devise new solutions to improve our world. I hope that you find that the innovative mindset is applicable to any endeavor, but maybe you'll learn a little bit about the specific content that each individual contributed to our world. And so that I don't have to water down the content and try to actually give more context to some of the innovators that I'm not so familiar with their background, I'll bring on some expert guests, and I'll introduce Nate in a moment. And I hope that you subscribe and it help us embark on a journey that puts more innovation in your life. So I met Nate Bachtel, our first guest on this podcast in 2017 when we were interviewing for MD PhD programs, and I met Nate at the Yale MD PhD interview actually. So now that it's almost 6 years into the program, we both decided to come here. What are you up to now?
Yeah, thanks so much for having me. I'm really happy to be here and excited to be able to talk with you today about an area that I'm really passionate about. So I'm currently in the MD PhD program at Yale, just like Brooks said. And for my PhD, I'm studying immunology, which is the study of how the immune system works. And in particular, for my PhD, I'm interested in allergies, specifically food allergies. And I'm interested in how allergies can alter the behavior of an organism, which I think is a kind of out there, but really interesting area to be able to study.
Where can people find the paper that you just published?
Yeah, so me and my co-first author, Esther Florsheim, both recently published a paper in Nature called Immune Sensing of Food Allergens Promotes Aversive Behavior. So if you're interested in reading a bit more about my work, you can find it there.
Awesome. And I'll put it into the show notes so that people can click on that link with one click.
Yeah, thanks, Brooks.
And I brought Nate on because we're focusing on the Nobel Prize winner, Elie Mechnikov, who in 1908, along with Paul Ehrlich, won the Nobel Prize in recognition of their work on immunity. So a very broad category that you won't see anymore. But I think it was pretty interesting. And before I sort of get into the background, my understanding is that his major innovation is really kind of understanding the role that immune cells play in helping us fight off microbial invaders. How would you say so?
Yeah, I would say that that's definitely true. And in particular, like you've alluded to, he was really important in kind of coining the field of cellular immunology. And within that field, he really made important discoveries on how your immune cells can actually engulf and eat bad foreign agents like bacteria or viruses. In addition, an area that I think we'll probably talk about a little bit less, but is equally interesting, he also realized that these same immune cells can engulf damaged cells or malfunctioning cells within the organism. So he was important in really coining this term phagocytosis, which is the process of cells eating something.
Thanks. Yeah, and this reminds me, we were talking a bit earlier that as I was reading the Nobel lecture, how he described this phenomenon reminded me so much of maybe going to the zoo, taking a bunch of breadcrumbs or something like that, throwing it into the pond. And all of a sudden you see the swarm of all of these fish or alligators, if you go to the one in Florida, that just go on and try to chomp on the food. So they sense that there's a new object in their environment and they go all rush up and try to eat it. So I don't know if that fits perfectly, but that's how I sort of envision this.
I think that's a great analogy. Both the swarming towards the new agent as well as the eating it and the digesting it afterwards.
Awesome. So his early life too, I think, is extremely interesting and kind of provides some insight into, that you don't often hear about, especially not scientists. But I'll read some excerpts about his early life. So he was broken by the loss of his wife to tuberculosis, who was so weak that she needed to be carried in a chair to the church for the wedding. And he was troubled by weak eyesight and heart troubles and also difficulties in the university. So at this time, he actually tried to take his own life by swallowing a large dose of opium. And so this suicide attempt was not successful. Obviously he went on to continue this, but it seemed like a really dark time for this guy. So I'm wondering sort of what attributes this experience might have helped shape his further identity. You actually read a little bit more about why he actually wasn't successful because of the dose of opium. Could you talk about that?
Yeah, exactly. So this was an extremely dark time in his life, and like Brooks said, his wife, is extremely sick and passed away. And at the same time, he was beginning to put forth some of these theories that were very opposing to the views in the field. And so I think those two things combined with potential inability to do experiments because his eyesight was failing really led him to make this choice. And he wasn't successful not because he didn't take enough opium, apparently, but actually because he took too much opium to the point where he ended up vomiting it all out, thank goodness. And this was probably the major contributor to him actually surviving that event, which I think is pretty remarkable. And it speaks to, after that event, you would potentially expect that he would have a new appreciation for life and some resilience. But as you'll speak about in a second, his life still, he had these repeated episodes of very, very difficult timing that made these suicide attempts kind of occur not just once.
Right. Yeah. And it actually is a very similar story, and it almost speaks to the time of what really was going on in the world, how people were succumbing to disease, how people were dying, because his second wife, whom he married several years later, he was with her for 5 years before she had a severe attack of typhoid fever. And though she didn't die, his health was still poor, and he again tried to take his own life. And this time he actually tried a different means. He inoculated himself, so basically gave himself relapsing fever to find out whether it was transmissible by blood. So once again, he's faced with this incredibly hard situation because we don't really understand a lot about infectious disease at this time. This is what is really killing young people, so I think it's very interesting, but he was driven again. He still hasn't found his hope yet to trying to take his own life.
Yeah, and how sad it is to think about that. One of the reasons why he chose this method was to not embarrass his wife who had passed away. And he kind of was using this as an opportunity to funnel his suffering into scientific discovery. But I think this just shows the state of kind of desperation that he was in at this time.
Yeah, and it makes me wonder if, he discovered sort of a new resilience, either about himself or about the human body in some way. Surely he didn't expect that he would survive these. We actually were talking earlier that, the decision to attempt suicide usually is actually made, after a long period of suffering or a long period of hopelessness without any help. But that actual decision is not often planned and long in advance. Usually it happens within a day or so. So it's a really, he was in a really, really dark place. And I think he had no real hope or purpose at this time that he felt until he sort of found a chance encounter with another professor, Klaus. So I'll just talk a little bit about this chance encounter where, in quote, he abandoned his pessimistic philosophy and determined to find further proof of his hypothesis. And this hypothesis, as Nate mentioned, he was actually working on some experiments that weren't really well received by the immunology field at the time. But he was able to discuss with Klaus, a professor of zoology, that suggested the term phagocyte, so the actual type of cell that eats these microorganisms or other objects. And if it weren't for his chance meeting with this guy, it'd be hard to tell whether or not he would have found hope or sort of a goal to pursue. And, what do you think the influence is that he actually met somebody else he could discuss these topics in science with?
Yeah, I think it was crucial for, his ability to find hope and move forward in the work that he did for a number of different reasons. One thing that we haven't talked about is that Mechnikov actually was not an immunologist from the get-go. And immunology, how we currently think about it, was very, very different back then. So he actually started out studying digestion. So how can single cell organisms eat other either organisms or particles to obtain food that they need to survive? And really, I think that this early work trying to understand digestion is what enabled him to come up with these kind of innovative new ideas in immunology. And I think it also speaks volumes that Klaus, the one person who was really able to kind of affirm his viewpoint and get him excited about his ideas and confident about his ideas, was also a zoologist. So these were both kind of outsiders of the immunology field, but together through a shared lens of viewing the world, they were able to really push forward these new ideas, which I think was crucial.
And I was beginning to smile at the end of this, because if you're not in academia, or if you're not a scientist, you might think that all scientists talk, and why wouldn't there be a universal understanding? But it's actually relatively rare for people in different fields to have communications, especially at the furthest edge of our understanding of a field. So it's kind of unique perspective that we'll talk about more, that he brought as an outsider to immunology. So it's almost like, I think that all business people should be talking, because you're all finance. Yeah, I'll have a finance background. It's similar in science, where usually the fields are kind of kept separately.
Yeah, exactly. And really, to move things forward the best, or to do the best job on any project, you need these experts who are thinking about very different aspects of the problem. And I think that this Nobel is like an amazing example of the importance of that.
Yeah, and you talked with me earlier about just the state of the immunology field. So kind of everybody was very interested in the common thoughts of there was sort of one way of thinking. Could you talk about Virchow? You were mentioning that.
Yeah, exactly. So Virchow was basically the father of modern pathology, which is the study of disease. And at the time of Virchow, what basically was thought was that inflammation, which often leads to a few different things, such as pain, reddening of the area that is infected or inflamed, as well as loss of function, and often swelling, was basically all thought of as a bad thing. So it was thought that basically inflammation was a reflection of something terrible going on in the organism. And so what was interesting was that Mechnikov's main contribution to this whole area was almost flipping that on its head. So often people were looking under the microscope, and they would see these immune cells kind of going around, swarming to these areas where there were, for instance, infections, so maybe a bacteria or something like that. And they would find, and pathologists had described this before he did, that some of these immune cells would be filled up with bacteria. But the pathologists at the time, viewing inflammation as this kind of negative process, thought it was actually that those bacteria were killing the immune cells, or that basically something was wrong. And I think Mechnikov flipped this idea on its head, as I mentioned earlier, because he was studying digestion. So he realized that maybe this process actually reflected an immune cell going and engulfing and eating and kind of digesting these harmful agents, and that this could be a system to prevent harm caused by these microorganisms.
Yeah. And I just, as you're saying, flipping it on it's so hard to try to come up with an idea. That there's probably been plenty of ideas that you've had, or any of us have had, that you're like, what if it was like this? The first person you tell would say, that doesn't make any sense. Right. So actually sticking to what you believe, and actually continuing to even think about it, because it's so easy to, well, that thought wasn't a good idea to my supervisor or to my friends, so I'm not going to think any more about it.
Yeah, exactly. And, I think it takes a lot of confidence to really be able to stand up for your viewpoint amidst all of this negativity from the outside, and to pursue that viewpoint and pursue experiments or angles of testing whether that viewpoint is correct or not, despite what the kind of dogma might be at the time.
Right. Yeah. So he seems like one of the more innovative, I think, of the Nobel Prizes. That this is the third one that we're doing, but I think that this is one that really you begin to appreciate the systems level change. So there's sort of incremental changes that, kind of follow a logical progression of something. And then there's system levels things that just require a new way of approaching and understanding a problem. It seems like he probably fits more into the systems level change in our understanding of the immune system.
Definitely.
And so we'll go into a few excerpts of the science and try to extrapolate a little bit on what was going on. Just one way I think that, is pretty interesting, that in order for him to even design his experiments, or basically how to approach the problem of the immune system, was really theoretical. So he starts with, because he realized he's at this point working on infectious diseases, and he thinks that these immune cells have a role in fighting them. So theoretically, he begins thinking, okay, so luckily for us, it is not only man and the higher animals that are subject to infectious diseases. These diseases existed on Earth long before the appearance of the human race, and few are the creatures which escape them. So he begins already saying, well, if there is an immune system, it's probably not unique to us humans. We know this because animals die. Like, everyone has observed other organisms being sick. Do you think that this is how immunology is sort of done? How we think about model organisms, how we think of how to test our questions, or how science is done in general? Or
Yeah. I think that this is actually a really wise viewpoint by Mechnikov, to think that all organisms are going to be faced with a similar problem of other organisms trying to use them for a food source in various different ways. In this case, he's recognizing the fact that even super small organisms that just have maybe one cell or even a few cells also undergo these problems of infections, just like, for instance, humans do. So is everything the same between the small organism and humans? Well, probably not. But there may be some very fundamental components that we can observe in these earlier organisms that are able to inform how we think about humans as well. And this viewpoint, I think, is really interesting, and it kind of fuses evolution theory, basically the idea that our immune system came from probably a very simple process involved in digestion, and that it kind of elaborated over time into being this way that we deal with and fight off infections. I think that this is a really cool way of thinking about and doing science.
Yeah. And it's, as you're talking, it reminds me of just the concept of first principles that you might hear in engineering or business or something, where if a phenomenon is going to be something that exists in our world at the largest, at the most fundamental level, you would like to begin studying that at the most fundamental level. And it seems like that's what he did here.
Exactly.
And that leads into, I'll just give an excerpt of one of the first experiments where he demonstrates the first stage of inflammation. So he actually takes some little thorns and puts the splinters into the bodies of bipinnaria. And then he says, the next day I could see a mass of moving cells surrounding the foreign bodies to form a thick cushion layer. And I believe this organism is see-through, so he could actually watch this without disrupting the organism, without dissecting it or cutting it open. So, what actually is he describing here when he says he puts splinters in the body? Is this one of the first examples of the cellular immune response?
Yeah, so it was known that, as I was talking about earlier, that cells do kind of infiltrate, and that you can find within the tissue itself, or within the infected area, that there are more immune cells there, and that some of them are full of these bacteria or other microorganisms. But what's different about this is that he actually went off and started doing these experiments and really was asking questions about, if I put, for instance, a red food dye, does that get ingested by these cells? And so he's really fundamentally looking at this through the lens of digestion. But as he's viewing through the microscope at these are actually starfish larvae, which is kind of a crazy thing that
Classic.
You won't really see in modern science, but I think shows his resourcefulness. Here he began to realize that maybe this process would also defend against foreign agents, not just this dye, which he was thinking about as digestion, but it could be, for instance, that a thorn that, like you said, was injected into this larva would be defended against by these cells. And you're right in saying that this idea of it being a defense rather than something that's going fundamentally wrong was really, I think, what this first experiment pointed out to him and gave him validation that these cells may not just be involved in digestion, may not just be bad for the organism, but may actually be a mounted response to help protect them.
Awesome. Yeah, and I just want to give a small excerpt is that these thorns actually came from a tangerine tree, which had been prepared as a Christmas tree for his children. So he just pulls out these random things in his environment and does, key observations in an experimental way. So as you mentioned, he's very resourceful, and he just was able to just grab whatever was around him and test his thoughts. And I'll then elaborate on the next piece of the science that you described. This might have been the first time that he understood the defense system of the organism. He actually goes on later to say, he's describing this hypothesis more fully. This is his research career. He's beginning to develop it and build on each experiment as a time. He says, disease would be a fight between the morbid agent, the microbe from the outside, and the mobile cells of the organism itself. Cure would come from the victory of the cells, and immunity would be the sign of their acting sufficiently to prevent the microbial onslaught. So I like this because he talks about it as if it's a battle, as if it's really a defense system. And, I think it's one of these elements of his way of thinking, and that everyone kind of can envision war and battle, and the different players, that there are different strategies that have been employed for thousands of years. So as he begins tackling a new problem, you wonder if his understanding of what war is might actually influence what he's discovering from the field of science.
Yeah, I totally agree. And I think a lot of Mechnikov's contributions that make him such like an amazing Nobel winner are really conceptual in a lot of ways. It's the questioning the fundamental nature of what people are saying. It's flipping it on its head. It's drawing analogies to assist developing the theories further and to kind of come up with new testable hypotheses. He's really a philosopher, I think, foremost. And of course, he's a wonderful scientist as well. But I think his ability to really think critically about these problems from a variety of perspectives is probably really aided him in his ability to make the contributions that he made.
Yeah. And now we can circle back to something you mentioned earlier. Just in being so innovative, he's going to have a lot of enemies. So at the time, a lot of scientists still kept to the old idea that the white blood cells were a bad thing. These are hostile to health. So, how do you go against an entire field really and push against this? So, I'm trying to imagine how this would even pan out today. I think obviously it happens, and we've actually seen probably some people that make very large claims, and now even journal articles can get retracted, for example. So you can publish a scientific experiment and then even get it through peer review, so we have other scientists read it and evaluate it. But eventually we realize there's a fundamental flaw, and then it can be removed from the record. So you wonder if that was happening at the time in any way, and people were wondering, okay, is this guy is he all talk? Is this bogus? He doesn't even understand the immune system. He's not from our field. You wonder what things he might have been up against,
Yeah, I think he was up against almost all of the scientific community at the time. And one of the wonderful things about science is that everyone can observe the same thing. And that's the most important part, right? Is to make sure that your observations are accurate. The stuff that you're presenting in your papers to the general public is accurate. But the same thing that people observe can be interpreted extremely differently. And I think that he's a great example of that, where people had been looking at similar things for a long time, but it really took him viewing it from a certain lens to move the field forward and come up with the necessary next steps to test whether this new theory of cellular immunity and phagocytosis was as important as he was making it out to be, whether it was beneficial as opposed to detrimental, for instance. So I think, as we talked about earlier, in his personal life, he had a lot of tragedies occur. You can imagine that being a real innovator it could have been totally possible that, none of his scientific contributions could be really appreciated till later on. And science in some ways is self-correcting. So hopefully someone would have been able to flip it on its head eventually and realize the important contributions that he made. But sometimes people are very ahead of their time. And he was definitely an example of that.
And it's interesting as you're talking too, I can't help but think how hard it is in general to keep an open mind when there's certain ideas that are just so different from anything you've heard about. These can be small, everyday sort of items or people from different levels. Like sometimes the best questions might come at a scientific talk from the most junior people, the people that actually weren't there for the last 50 years of science, because they have the most open mind at that time. And almost as you learn more, it probably becomes harder and harder to keep an open mind unless you practice it. So I think that not that he necessarily had an open mind, but the field that he was up against seemed like not many of them had open minds. And the only person that he seemed to have supported his ideas early on was someone from his field.
Yeah, exactly. It's from another outsider who was like, this is a really cool idea. You should definitely pursue this further. And that's the sort of validation that he needed to really take it to the next level. Yeah. It's interesting.
And we don't have to go too much into the science, but I just wanted to discuss a little bit where in his Nobel lecture, the other theories that are going on, non-cellular theories of how the immune system worked, he ends up acknowledging that probably both theories are probably correct. And we understand today that, yes, there's, multiple contributions. But as he's putting forth this cellular theory and everyone working on the other theory, I wonder, did he have to double down on having a one-sided view so that his point was heard? You wonder maybe how much do you think he believed that the other theory was real? Do you think that he thought that everything was explained by cellular, or he probably had an open mind to some extent, but needed to make sure that his voice was heard?
Yeah, so as a little bit of context here, this Nobel Prize was awarded to Mechnikov for this idea of phagocytosis and cellular immunity, as well as Paul Ehrlich, who really pushed forward the idea of humoral immunity, or immunity that can be found in non-cellular components such as antibodies, as well as something called the complement system, which I won't describe in great detail today. And so I think basically since this idea of humoral immunity, these chemical factors floating in the blood and things like this was such a accepted phenomenon at the time, people almost had narrowed vision where they couldn't see any other explanations for what they're studying. And so in order to combat that, when Mechnikov began realizing that this cellular component of the immune system was crucial for mounting a proper immune response against these microorganisms, I think he for sure had to sell it. He had to sell it. He had to argue for it. He had to even frame some of the experiments that were done in this humoral immune field as being actually mediated through his phagocytes, right, for instance. And it's really interesting, hearing it kind of, at the time, it was kind of two opposing theories of immunity, but now we understand that they work in synchrony to really perform the proper function. And just as an example, we talked about these antibodies, and we've talked about phagocytes, which help engulf microorganisms. Well, it turns out that the phagocytes have things that bind to the antibodies, and that antibodies help them find the things to engulf and to engulf them more efficiently. So I think he definitely had to stand up for his viewpoint. And in his Nobel lecture that we read, it was very clear that he was trying to make an argument for this cellular immunology field. But it's really amazing how they both kind of came together. And now it really highlights how the immune system works so efficiently.
Yeah. Yeah, and I wanted to talk a little bit about the medical applications today and sort of the relevance today of any of this stuff. Actually, in the lecture, it's actually separate from the other lectures that I've read. Elie Mechnikov actually attempts to say how this will be useful to medicine. So I think it's interesting. Many basic scientists, so these might be people with PhDs that actually never see patients or never go to medical school. It's often talked about that, there's a large disconnect from the actual medical applications. And sometimes there is. But I think it's rare for a basic scientist like Mechnikov to actually say, I actually think this will be useful for a lot of applications in medicine. And when you apply for government grants, now you actually have to say that even when you're doing very basic research now. So I don't know if you always had to. It doesn't seem that way. Describe why it would be important for medicine. I've even seen other Nobel Prize winners say something like, it will be great to one day realize the practical benefits of my work, right? And exactly. So not everyone actually has thought about this deeply, but I was impressed by that aspect.
Yeah, and just as one more point related to that, it's not always clear how it will be useful. Mm-hmm. So, if you think about the story that we've just told you about today, this is a zoologist who is trying to figure out how very small organisms digest things. And through that, which is tried sending that to the NIH, they're not going to fund it at all. But through that, he basically discovered this super fundamental thing about the immune system. Mm-hmm. So basic science, for the sake of basic science, I think makes a ton of sense when viewed through that lens. But at the same time, like you're saying, once you know a bit better what's going on, being able to think about what its practical applications are is very important as well.
Yeah. And he talks it's funny because it's actually a common discussion that you see. And that's why there's so many different funding institutions for where you can get your dollars from. And that's, in part why people try to get private funding and things like that, where different organizations either the people, the investigator themselves, and you can imagine just like if you're founding a company and you want to get venture capital funding, usually you will invest in who you think is going to be the best founder, regardless of what idea they pitch you. There's certain attributes about a character that you might invest in. And so that's a unique funding model that exists maybe through Howard Hughes Medical Institute, for example. But it's interesting just thinking how science is funded in general. It's most often about a particular project if you go through the National Institutes of Health. But there's other avenues for different ways of funding science that might allow you to remove the direct medical application a little bit. But he talks about using these immune cells in the blood as a biomarker. He says, it leads one to conclude that the degree of phagocytosis can be used in medical prognosis. And funny enough, we do this all the time. Let's get a CBC so we can look to see if they have a leukocytosis, right?
Yeah, exactly. You can do a gram stain to see if the bacteria are inside the cells and what cells they're inside of. There's a lot of, he was totally kind of spot on with regards to it being able to predict certain infectious diseases, for instance, and how severe or not severe they were.
Yeah, and he talks also about that in order to prepare for surgery, I think it seems more fundamentally that we're realizing that these bacteria can cause really severe infections. And this is, probably beginning to have a much better appreciation of the real importance of that. Having seen so many young people succumb to certain infectious diseases, right? It's probably really said, he goes on later in his life to sort of champion a lot of philosophies and a lot of theories. But I think that, realizing that we should try to minimize the onslaught that we get from these infectious diseases and improve. He talks actually about enhancing the immune system. So, and that's something that maybe we could discuss today. I think that's, immunotherapies for cancer. I don't really know in the field of infectious disease. How do you think about what he meant by enhancing the immune response?
Yeah, so it's pretty interesting now because, like you said, Brooks, although most of what we've talked about today has been through this infectious disease lens, and I think the reason for that is because the Nobel lecture that he gave was given during a time where infectious diseases were so prevalent and so many people were dying from infectious diseases. So he's really selling it to the audience. What we've realized over time, though, is that this process of phagocytosis is actually fundamental to pretty much all aspects of our biology, and that as we have new antibiotics, so we don't have as many infectious diseases, we tend to live longer. And so now the problems that we suffer from are not as much infectious diseases, for the most part. There are exceptions, obviously, but they're from these other diseases that are related to basically breakdown of tissues over time. So one example of this could be, for instance, Alzheimer's disease, and cancer could also be viewed as a similar breakdown. And what we're realizing is that this phagocytosis that happens during infectious diseases is also really important for clearing out malfunctioning cells. So it's crucial for clearing out, for instance, plaques in the brain from Alzheimer's. It's crucial for identifying cells that are going to become cancerous and clearing out those precancerous cells. And people are now realizing that cancers even put signals on their surface that say, hey, don't eat me. And so these are kind of emerging immunotherapies that people are using to try to manipulate the immune response to cure things such as Alzheimer's or cancer. And there are many other areas independent of infectious disease that Mechnikov's work is inspiring today. And, this is an important point because immunology right now is kind of focusing very heavily now on these non-infectious diseases. That's a huge area that the field is moving towards. And I think it's amazing that Mechnikov, back over 100 years ago, already was thinking that these would be some applications that this system would defend against.
Yeah, and you talking reminds me of more context, right? In 1900, I think 6 of the 10 leading causes of death were infectious diseases in the United States. And today, it's, I think, maybe 1 or 2, other than COVID-19 in 2020 and 2021, there's maybe 1 other infectious disease. There might be 9 on, respiratory illness in general might have been it. But 86% of healthcare spending today is spent on managing chronic diseases. So now, I wonder if that just reflects the state of science and the state of how we're framing problems today. We're beginning to realize that, maybe then they were realizing these are really the problems that people are dying from.
Yeah.
And what people are suffering from today are these these chronic diseases.
Yeah, and it's interesting. I mean, even taken apart from this phagocytosis that we've been talking about, Mechnikov was so forward-thinking that a few of the other ideas that he came up with at the time were gerontology, or aging as a field of science. That's kind of makes sense now that we viewed it through the lens of phagocytosis. And then the other is actually microbiota. So he was a huge proponent of probiotics, and basically had some papers on the benefits of, for instance, yogurt for gut health and longevity. So for a long time, he was actually, before people realized the phagocytosis theory was so important, people knew him as the guy who inspired like all of Europe to eat a ton of yogurt and stuff like that. So this was a guy who was very forward thinking to the point of almost being, I would say at the time, probably some people ridiculed him for being so forward thinking.
Absolutely. And I remember reading in some other text that he had written that he believed everyone should be able to live to 120. Humans should have the intrinsic ability to live to 120 if it weren't for the decaying of cells and things like that. So he's just began thinking about these problems that are pretty hot now, that especially the yogurt and the probiotics, and like kombucha. I don't remember when we were younger, like being sold in that many stores, and now you see it everywhere.
Exactly. It's really crazy.
So it's just in that sort of thing. He might have even had a hard time finding colleagues that would support him on those things. You're like, hey, you're stepping out of your field now.
Yeah, exactly.
But it's interesting. I think that moving into some of the qualities about him that facilitated his success is, you mentioned earlier, this philosophical worldview applied to biology. He's really able to create frameworks to understand things at, 1,000-foot view. How do you think that helped him form questions and guided his science?
Yeah, I think that philosophy definitely guided a lot of what he did. From the very get-go, philosophically realizing and viewing digestion through the view of evolution led him to study these much smaller organisms, even single cellular organisms, for instance. And I think that led him to, even take a further step back and question, is this process of digestion the same as this process of immunity? And so I think he was one of these people who is able to really, do the experiments when needed, but always take a step back and think big picture. What could this mean, actually? Instead of narrowing in on very specific data points or results, taking a step back and thinking, what is actually going on here? What could this mean? And he was amazing at doing that and kind of using that to push the boundaries of what's known and test whether his viewpoints were accurate or not. Is inflammation beneficial or detrimental? Can we test that? You can't do that until someone proposes that inflammation could be beneficial.
Right.
So he was great at thinking about these problems from a ton of different angles. And like Brooks said, putting them in this framework and kind of forming a worldview based on them. Yeah.
And this outside perspective he brings, I mean, that's something that, I think you'll see a few places. I think it's increased, the importance of which is becoming increasingly common to try to get together these different backgrounds, these different perspectives in order to begin thinking about things differently, because only a junior person who might have been very well trained in another field, might ask the most fundamental basic question of another field, right? And it's almost like if there is no cross-disciplinary thing that you could just go a single track mind, there would be no way of thinking outside of the box at all.
Yeah, and all of these systems interact with one another. You have one organism at the end of the day, right?
Yeah.
And so how is it that these different systems within an organism interact? And I think that is an area that is gaining a lot of popularity, is how do these different kind of systems that we've thought about as being separate talk with one another. And similarly, in other areas too, like we spoke about at the very beginning of the episode, I think having as many diverse viewpoints on the problem will lead to the most effective solutions at the end of the day.
Yeah. And he was exceptional, I think, at simplifying problems. That's another, just stepping further and further and further back. What assumption needs to be true in order for this to be? Questioning assumptions, from the earliest fundamental components, right? How can I simplify it so that I have a true understanding of what actually needs to have been true, so then I can understand what the next logical step might be. And I think that's a very hard thing, especially when you enter a new field, or you're starting out a research career or something, or any career for that matter. It's kind of hard to take a real step back and simplify it rather than kind of pick up where you left off from a mentor or another project or something like that, because that's what you're in the mindset of doing. But being able to actually simplify the problem to the most fundamental level so that you can understand where can you make a big improvement on a system, I think he did a great job at. I mean, working with these larvae in his backyard, I think is a way for him to say, if it exists in these organisms, it probably will also in guinea pigs and maybe also with us.
Exactly. And now, one thing that kind of stems off of that idea is being able to study this phenomenon in many different systems. And Brooks and I were talking earlier, and we were talking about now how science is done. Obviously, there are certain scientific standards that are extremely rigorous, such as, you need to use the same mouse strain for everything that you do, or you need to, use the exact same fruit fly. There's a lot of kind of control checkpoints that are put into place so that you can make the most correct assumptions about what is causing the effect that you're studying. But I think what Mechnikov does well is he almost does the opposite. He says, look at me studying
it all.
Yeah, he says, look at me studying these like amoeba, look at me studying these starfish larvae, look at me studying these guinea pigs, and all the way up, to human biopsies and things like this. And he's able to see the same phenomenon all across those. And I think that really probably speaks to the importance of phenomena that are discovered that way. Mm-hmm. If it's so fundamental that you can see it happening amidst all of that chaos, it's probably going to be an important feature of the system.
And the wild thing that we discussed this earlier is being able to actually pick up on what those observations mean. There's probably how many phenomena are going on in front of us every single day that are related to something else or have a same sort of relationship between two components that might help us understand some new breakthrough. But it just takes someone to put that all together.
Exactly.
It's pretty wild. Yeah, and this ability to move through model organisms very quickly. I mean, for example, if you want to do a new experiment, when I was working on a sepsis project during my PhD, and we wanted to try different strains of bacteria and everything, and you need to submit so many different forms to so many different people, you need to get your lab approved. It takes months to do something that he probably did in hours, right? So
yeah, exactly.
It's interesting. Not that it's a bad thing, right? It's just it's a trade-off in sort of how he was able to do science over 100 years ago versus what we can do today.
Yeah, and it almost feels like currently there's such an infrastructure to be able to tackle certain problems, and we've gotten really good at using certain tools to attack those problems. And reading about someone like Mechnikov makes me feel like maybe we need to diversify the models that we're using. Maybe we need to try to think about what other models would benefit us. For instance, are amoeba a good model to study? For certain questions, they could be the best model. Are shrews or some other mammal more useful to study for certain things? And really, right now, you don't really see the development of tools in those areas. You see that everyone's kind of studying the same mouse strains and things like this, which is useful, obviously, for making controlled conclusions. But it makes you wonder how much we're missing by not looking at the breadth of what's out there.
Yeah, that reminds me, I remember watching one of Marcelo Dietrich's either postdocs or graduate students give a presentation on the armadillos that they were working on for a particular project. But one of the main issues is there was no genome sequence for the armadillo at the time. So actually doing the standard techniques that are used kind of every day in other laboratories in mice or other organisms are almost impossible to do, and they're limited in it. It would demand a group that goes on that in order to keep up with all the technology, expensive technologies that we use today. I was just thinking, like, I wonder if anyone has a mass spectrometer in their own house, this $200,000 piece of equipment that you can just pick things up in the backyard and analyze chemical composition. But there's a lot of things today that, make it, with technological advances, like you're saying, are definitely kind of limiting in some ways. But hopefully that enforces creativity, like we're seeing in a lot of these early Nobel Prize winners. He didn't have tons of funding, tons of resources. It's just, let me start working with what I can do, what I think is important, and just begin working at that.
Totally agree. Yeah, I think it's a lesson for all of us in terms of how to make the most out of our surroundings.
And so I'll finish with a few lessons that we can discuss. I mean, one thing that he teaches that I try to remind myself, especially even for this podcast, or, that's a real challenge, or when giving a presentation, is to simplify. Take one problem at a time, identify the key players that are essential to that problem, and determine their relationships. So I think that's an example that he did really well that I'd like to take. I mean, how hard is it to simplify things, right? I mean, especially complex topics or nitty-gritty details, it becomes very challenging to take a step back and realize what is the most basic component, the essential components.
Yeah, and there's a difference between properly simplifying and oversimplifying, right? So in his lecture, he actually mentions, too, that certain conclusions that are drawn from cells in a dish may not be accurate ones, right? And so it's really simplifying to the proper extent to be able to most effectively tackle the problem in question accurately without oversimplifying such that you're losing important information about what's actually going on.
Totally. And that's another lesson that I pulled out in that vein, is choosing your problem-solving approach wisely. There might be multiple approaches, like that in vitro, or looking at cells in a dish was a common approach that a lot of people were making fundamental discoveries with. That doesn't mean it's the right approach for every problem. So I think that he did a great job in saying, why would this approach be the best thing for the problem I'm trying to go? And that's when he described, infectious diseases have been around for millennia, right? So this probably exists in another setting. So let me just come up with whatever way allows me to test that.
Yeah. And is there a system, since it has been around for so long, mammals are not really see-through. And so he realized that he could take advantage of these organisms that were see-through in order to study this in a living situation.
Mm-hmm.
And I think that was crucial for him to be able to convincingly study these things.
Yeah, I think I now when you mention it, I remember him saying like, and now that I have to move to higher organism, everything gets a little messier.
Yeah,
you know,
exactly.
you have to take out their organs and their cells and all that. And he's like, it's just way too complicated.
Yeah, and you're trying to interpret what's going on outside of the organism as opposed to while it's actually in the organism.
Right.
And that's still a problem that we deal with in biomedical research today.
Yeah. And just another sort of lesson in tying in his philosophical mindset, his sort of systems thinking. So is his way of chasing analogies. So this is where I just liked seeing him constantly referring to the immune system versus microbes as a war or a battle. Sort of a more simply understood, well, at that time, thousands of years, people have understood war and battle and the key players of those components, the relationship that a general might have. So maybe that was the brain. I forget. I think some people talked about the nervous system. But you can imagine just abstracting these components of war that maybe he was able to do with the immune system. And you can imagine, there's so many ways that, we can begin thinking about new problems by actually using something we already understand very well.
Yeah, I totally agree with that. And this really speaks to him being a philosopher more than anything else. There are some scientists who only are able or only choose to make incremental contributions to what's already known because they feel like taking a step back and abstracting, they may be wrong. But Mechnikov looks at it differently. He says, well, if I take a step back and I broaden my thinking and I rigorously, philosophically, and logically test that thinking, that's still a way that we can move things forward in a very major conceptual, innovative way.
Yeah, and this ties in, when you're doing something like that in any endeavor, trying to start a business that people think will fail, and you've heard plenty of examples of that, you need to find someone that believes in you. Find someone that you can actually discuss to the fullest extent what you're thinking, what those ideas are. This was that guy Klaus in the very beginning, this sort of chance encounter that he was able to find someone that believes in you. And I talk in the second episode about actually nurturing and fostering the relationships that you can discuss the fringes of our understanding with. And it seems like Mechnikov certainly needed that push, especially in his darkest times. It seemed like it was only that sort of fundamental conversation that he had with Klaus that kind of gave him a noble goal to pursue and really kind of got him to feel like he had a purpose in life.
Yeah, and some of the most, I think, innovative ideas come from when you're thinking without any filters. So often, I think, what we do in the lab that I'm in and what other, even outside of science, other companies encourage is just open brainstorming. Mm-hmm. Some of the ideas that you have when you're brainstorming openly may be terrible ideas, right? And that's fine. The most important thing is that it allows you to think about all of the possibilities, and you may think of ideas that you never would have thought of otherwise from removing those filters. And like you're saying, having a mentor like Klaus really allows you to do that. You're with someone that you feel comfortable enough with. You're fine being wrong. You're just trying to have open discussions about the possibilities and to discuss new theories. And it takes sometimes an outsider, like a zoologist, for instance, in this case, to really find the right perspective of thinking about a problem.
Mm-hmm. And I'll just summarize these sort of last four lessons, right? So simplify, simplify, simplify. Use system thinking and chase analogies. Choose your problem-solving approach wisely. And find someone that believes in you. So, Nate, it's been a pleasure having you on here. I hope that you come back again and so we can do this again. It's been really fun.
Yeah, thanks so much for having me, Brooks. I've had a great time too.
Sweet. Now, go innovate.